This first part of Chapter 7 will focus on the structure of the genome, which is made of nucleic acid, specifically DNA. DNA is made of a smaller component called nucleotides. If you remember from Chapter 2, nucleotides consist of a sugar, a 5-carbon sugar. In the case of DNA, is a deoxyribose sugar with 5 carbons, 1, 2, 3, 4, and the 5th carbon is outside the ring. To one side of the sugar, attached by a strong covalent bond to carbon number 1, we find a nitrogenous base. A nitrogenous base is a ring of carbons and nitrogens. To the other side of the sugar, attached by a strong covalent bond to carbon number 5, there is a phosphate group. DNA is made when carbon number 3 of one nucleotide forms a strong covalent bond with the phosphate group of another nucleotide. Carbon number 5 of the sugar is attached to the phosphate group, which in turn is attached via a covalent bond to carbon number 3 of another nucleotide. Thus, a sugar phosphate, sugar phosphate, sugar phosphate is created. This represents the backbone of the DNA molecule. Notice that the nitrogenous bases attached to carbon number 1 remain to one side of the molecule. DNA in cells is a double-stranded helix. The blue ribbon seen here, represents the phosphate sugar backbone of the molecule. The nitrogenous bases of one of the DNA strands face the nitrogenous bases of the other strand. The nitrogenous bases facing each other bind to each other via weak hydrogen bonds. The two strands of DNA twist on each other, creating a double helix. So remember the nitrogenous bases attached to carbon number 1 of the pentose sugar. Remember the nitrogenous bases can be of two types, pyrimidines or purines. There are two types of pyrimidines found in DNA, thymine or cytosine. Two types of purines found in DNA are guanine and adenine. The nitrogenous basis of one strand forms weak hydrogen bonds with the nitrogenous basis of the opposite strand. However, the bases do not pair at random. Guanines always pair with cytosine, adenines always pair with thymine. Remember, thymine is present in DNA molecules only. RNA molecules have uracil instead of thymines. So thus a purine, guanine, always pairs with a pyrimidine, cytosine. A purine, adenine, always pairs with a pyrimidine, thymine. Also notice the difference between purines and pyrimidines. Purines are two nitrogen rings, or have two nitrogen rings, whereas pyrimidines have one nitrogen ring. Now DNA is a double helix, and we have a sugar phosphate backbone of one helix, and the nitrogenous bases of the DNA molecule to one side. Notice that on one strand, the 5 carbon is the last carbon unattached to another nucleotide in one strand of DNA. On the opposite end of the same strand, The 3 carbon is the last unattached to another nucleotide carbon. The opposite strand, if it faces in the same direction, 5 carbon to 3 carbon, notice that the nitrogenous bases will be attached to the carbon number 1, and they will not be facing the nitrogenous bases of the opposite strand. That represents a problem because the nitrogenous bases are supposed to pair with each other. In order for the 2 strands to be able to pair their nitrogenous bases with each other, one molecule has to be reversed upside down, so as now it is running in the opposite direction, 3 carbon to 5 carbon versus 5 carbon to 3 carbon. Now the nitrogenous bases are facing each other, and weak hydrogen bonds can form among them. Thus, the 2 DNA strands are said to be antiparallel. The 2 DNA strands in a DNA helix are antiparallel. One strand runs 5 to 3, the other strand runs 3 to 5. 3 and 5 representing the last unattached carbons on the strand. Thus the nitrogenous bases are facing each other, and they can base pair, forming weak hydrogen bonds. DNA strands are antiparallel. They're also complementary strands. That means that they are not identical, but they do complement each other. If one strand has a nitrogenous base sequence A, G, T, A, C, G, the opposite strand will have the complementary sequence T, C, A, T, G, C. Notice that having one strand can give us the needed nitrogenous base sequence to code for the opposite complementary strand. In prokaryotic cells, DNA, the genome of the cell, is found as a single chromosome in the nucleoid of the cell. The nucleoid is a discrete area of the cytoplasm of the cell. Prokaryotes also contain DNA in the form of plasmids. Plasmids are small circular DNA molecules that carry genes that are non-essential to the survival of the cell. Plasmids replicate independently from the chromosomal DNA. They carry information required for their own replication. They carry information for one or a few more traits which may give the cell survival advantage, but they are not essential for the survival of the cell. In other words, a cell could survive without plasmids. When a prokaryotic cell divides by binary fission, first its DNA divides, then the cytoplasm of the cell divides, creating 2 identical cells. Notice that some prokaryotes may have 2 instead of 1 DNA molecule. The DNA in prokaryotes is a circular DNA molecule with 1 origin of replication. The origin of replication is the place from which replication of the DNA molecule will proceed until 2 identical molecules have been synthesized. Plasmids carry genes that might give the cell survival advantages. We will look at 2 types of plasmids in Chapter 8, fertility factors and resistant factors. This is a scanning electromicrograph of a cell, a prokaryotic bacterial cell, whose chromosomal DNA has spilled. This is a huge circular molecule. Doesn't look like much a circle, but it is. Also, its tremendous size gives you an idea of how well that molecule must be packaged inside the cell in order to fit. These smaller circular pieces of DNA are plasmids. Eukaryotic DNA, on the other hand, is found inside a membrane-bound nucleus. It consists of several linear molecules of DNA. When the cell is not dividing, these linear molecules of DNA appear as diffuse structures called chromatin. When the cell divides, these diffuse DNA molecules organize themselves into discrete entities called chromosomes. This is diffuse chromatin. Here are organized linear eukaryotic DNA. Structures are called chromosomes. Eukaryotic DNA consists of several pieces of linear DNA molecules. Each linear DNA molecule has several origins of replication. Origins of replication is the place where DNA replication proceeds. In this case, we have several bubbles of replication, with the blue molecules being the templates from which the red molecules are the newly replicated DNA strands. These multiple origins of replication assures that the DNA will replicate much faster than if there was only one origin of replication. When linear eukaryotic DNA is being packaged by proteins called histones, when the cell is ready to divide, it is packaged very tightly and creates these discrete structures called chromosomes. Eukaryotic cells also have DNA inside two organelles, the mitochondria and the chloroplasts. Remember, chloroplasts are found only in photosynthetic cells. So inside the mitochondria, inside the matrix of the mitochondria, there are circular pieces of DNA which carry genes to enzymes needed for mitochondrial function. So this mitochondrial DNA resembles prokaryotic chromosomes, prokaryotic DNA. At this point, you should be able to compare and contrast the genomes of prokaryotes versus eukaryotes. Which one has circular DNA molecules, which one has linear DNA molecules. You should know the location of each. You should know the origins of replication found in each. Which kinds of eukaryotes have plasmids. Obviously, pretty much all prokaryotes have plasmids. Also, the significance of the mitochondrial DNA being similar to prokaryotic DNA. It may be argued that mitochondrial organelles may have been at one point prokaryotic cells that were engulfed by an ancient eukaryotic cell and eventually became an organelle. So that is kinds of questions that you should be able to answer at this point.